from, but where are you currently located? And the hosts and panelists can't vote, but everybody else is welcome to do so. And it's very quick, so answer fast, because Fran is going to put up the results very soon. And we have 42% from North America, 24% from South America, 2% from Mesoamerica in between, 17% are in Europe, 8% are in the Caribbean, 3% in the Pacific Islands or Hawaii, and 3% in Asia. 1% is from Australia and New Zealand, and 2% are from somewhere else in the world, maybe Asia or the Middle East. Thank you very much. We'll close that poll. And here comes the second quick poll of which sector do you work in? And let's see how that poll is actually, how the question is actually posed on that poll. Is it coming up? There it is.
That's the wrong one.
That's the wrong one. Well, this is the first session, and I'm the least experienced about doing this, so I'm glad I'm not responsible for that technical end of the program. I can see that the chat is filling up nicely, and that's good. We want to hear from everyone, at least with telling us who you are and your contact information, in case you see somebody else or you hear from somebody else that you'd like to share information with later on. What sector do you work in? Here we go, the second poll. Do you consider yourself an academic, a student? Are you in conservation, management, government, tourism, education, industry, or any other occupation? And you have a minute to answer that question. And then Fran will put up the answers. And there we go. 31% in academia, students are 36%, more than any other group. Conservation and management is pretty good, well represented, that's 13%. Government, there's a lot of government people here, 13% also. Tourism, education, industry are all less than 5%, as are people in other occupations. So the poll is now closed, and we will get to today's presentations. This afternoon, we will be first listening to Drs. Esther Peters and Jennifer Salerno, both on the faculty at George Mason University out in Northern Virginia, outside of Washington, D.C. Esther is a renowned coral histopathologist over here in the Caribbean, and Jen works with her on the histopathology of the stony coral tissue loss disease that is now ravaging the Caribbean as well as Florida reefs. And in addition to that, she also engages in science communication and interdisciplinary work at the intersection of science, policy, and diplomacy. Esther will speak first, and then she will be followed by Jen Salerno. So could we please have Esther's and Jen's screen share so we can look at their presentation.
Thank you, Judy. Is everything set?
Looks good here.
Very good. Okay. Hello, everyone. I'll start where this has been the story of my life. Until about 1970, coral reefs were considered pristine. All their organisms were thriving. When I began my master's research, Phil Dustan, who is giving our second plenary talk today, was documenting the extensive elkhorn coral thickets at Carysford Reef in the Florida Keys. I studied the effects of number 2 fuel oil on corals as drilling and shipping oil were becoming concerns. Then for my dissertation research, I switched to study the effects of sedimentation and the second reported disease of corals, white band disease. When I got my PhD, Phil was discovering that more corals on Carysford Reef were dying from this and other diseases, as you can see in this 1985 frame on the slide. And unfortunately, corals on the reefs all around the tropical Western Atlantic started dying as well. Only more recently have we realized that their demise occurred during a period of increasing seawater temperatures and decreasing seawater pH and other factors. We have now seen 50 years of continuing coral disease outbreaks, leading to numerous coral mortalities, loss of the skeletal accretion that provides habitat for reef organisms and shoreline protection, loss of reproduction and larval settlement to renew the coral populations, and ultimately we have food web destruction. From the first reports of black band and white band diseases, shown on the left, a variety of disease outbreaks have been documented, including coral bleaching, black band disease, white plague, white pox, yellow band disease, and dark spots disease. Most of these diseases have been recognized to occur globally. In 2014, corals on the reefs off South Florida began to die from what has been termed stony coral tissue loss disease. And this disease has spread north and south along the coast, almost to the dry Tortugas, and has also affected corals on distant Caribbean reefs, including Jamaica, Mexico, Dominican Republic, Turks and Caicos, Puerto Rico, the US and British Virgin Islands, the Bahamas, Belize, St. Martin, and St. Eustatius. And yet, although a few pathogenic microorganisms, bacteria, have been identified and shown to be causing some of these diseases, we still have many questions about what is causing them. To obtain a disease diagnosis requires recognizing the earliest stages of the disease, what the lesions look like, what abnormalities in tissue structure might be associated with changing behavior and loss of normal metabolic functions. We also need to investigate the situation in the victims because we now know that both biotic and abiotic factors, usually a combination of anthropogenic and natural stressors now, can cause mortalities and morbidities. Infectious viruses and bacteria, as well as chemical contaminants and suspended particulate matter, and can all be pathogens. The situation in the ocean can change rapidly with the next tide or storm, but often the collection of observations and then samples have been delayed as agencies figured out who can do analyses and issue permits for coral sample collection. With delays, we are now, excuse me, I did not mean to go there, that we now have issues where we're going to lose the etiologic agents and we won't be able to identify them, and that compromises the diagnosis. I started to study coral diseases using the same methods that anatomic pathologists in hospitals use. Fixing a sample in a formaldehyde-based solution to preserve the cellular structures, embedding decalcified tissue in paraffin, then cutting off thin sections, mounting them on microscope slides, and staining them. By studying the sections with a light microscope, we can learn about their condition and take photomicrographs for further analysis. Tissue changes that affect vital functions can be present well before they are visually evident. Thus, an apparently healthy coral may not be in good condition microscopically. As seen in the images on the left, as well as the right side of the slide, Corals can have a variety of cellular changes, bacteria, fungi, or protozoa, altering their cells and tissues, despite being visually normal when they were collected. And the two images in the middle were collected from bleached corals. And many people would just say that the corals had expelled the symbiotic algae or zooxanthellae, but here histology revealed different mechanisms of leaching and tissue damage in these cases. One in which just the zooxanthellae died within the tissues, and another in which the gastrodermis is just dropping away from the epidermis. And there may be others. After bacteria were identified and the earliest recognized coral diseases, more microbiologists joined the investigation. This very incomplete synopsis shows a number of studies recording bacteria associated with or causing coral diseases, as confirmed by Koch's postulates. The first investigations were limited to culturing bacteria from ground tissue, skeletal samples, or mucus collected with syringes or swabs. However, most bacteria, we know now, cannot be cultured. And while the incorporation of molecular methods has somewhat helped us to circumvent this issue, it has also shifted our understanding of disease processes in diverse organisms and our reliance on Koch's postulates.
So one of the major barriers complicating our ability to determine coral disease etiologies is our inability to fulfill Koch's postulates, or the criteria that have traditionally been used over the past 130 years to establish a causative relationship between a microorganism and a disease. As a reminder, these postulates stipulate the suspect microorganism must be present in all cases of disease, not in healthy individuals, must be isolated in pure culture from diseased organisms, and then cause disease when introduced into a new susceptible host. It then must be re-isolated from the inoculated host and identical to the original isolate. However, there are major pitfalls with this approach that has prevented us from making progress with determining disease etiologies. First of all, the vast majority of microorganisms have yet to be cultured, preventing the fulfillment of the second postulate. Furthermore, with our exploration of host-associated microbiomes using molecular techniques, our understanding of the roles of microbes in host health and disease have shifted, and the traditional Koch's postulates, which state that one pathogen leads to one disease, are no longer valid. For example, polymicrobial diseases, or diseases caused by more than a single type or species of pathogen, like black band disease in corals, as well as dysbiotic diseases, are those resulting from a shift in the microbial composition or function and resulting in an atypical host response, do not fulfill Koch's postulates. Therefore, a more complete and complex interpretation of Koch's postulates has been called for to better understand and explain these diseases. In the late 1980s, the postulates were modified as molecular Koch's postulates to focus more on identifying a gene that could cause a microorganism to be pathogenic and lead to a disease, rather than relying on the ability to isolate a particular pathogen. This approach helps to explain infections caused by intracellular pathogens, which cannot be cultured outside of host cells, but also pathogenic strains of otherwise benign microorganisms. And although this was a good start, there are still limitations, including that some genes cannot be manipulated and some diseases do not have suitable animal models. So in 1996, Fredericks and Relman expanded molecular Koch's postulates to include sequence-based taxonomic identification of microbial pathogens for consideration. And then more recently, ecological Koch's postulates have been proposed, which look at the disease process from a whole ecosystem perspective to include associated microbiota, genetic makeup of the host, nutrition, age, as well as abiotic factors and environmental stressors, which all form an entity that can ultimately lead to disease. And these modifications are more in line with our current understanding of the role of microbiomes in host health and disease, as well as the molecular methodology increasingly being used to study them. So over the past several decades, we've observed a rapid increase in molecular-based microbiome studies as a result of advancing sequencing technology and decreasing cost. And of course, the same has been observed in the subfield of coral microbiology, with an increase of 16S ribosomal RNA gene-based coral microbiome studies over time, as shown here. And these studies have yielded an abundance of important information that has significantly advanced our understanding of coral microbiomes. For example, we have a better understanding of the types of microbes associated with corals under disease and non-disease conditions, and their potential functional roles with respect to the coral host. We're also able to use molecular methods to monitor dysbiotic and functional shifts in associated microbiomes in response to stressors, inching us closer to unraveling coral disease processes. However, there's still notable limitations, and despite the increase in molecular-based studies, we still have some pretty significant knowledge gaps to address. Some specific limitations of using molecular methods alone to investigate coral disease include the following. First, we tend to lose the spatial organization of microorganisms to the host tissue and cells at a scale that's relevant to both, and we cannot directly observe microbe-host interactions using those methods. And although we are improving in this regard with programs like the Earth Microbiome Project, there's still somewhat of a lack of standardized methodology being implemented in the field. We know that using different DNA extraction protocols and PCR primers can impact microbiome detection in downstream analyses. And despite the growing body of research, we still have not determined the majority of etiologies for most coral diseases. So what are we missing? Well, it's important to acknowledge that increased culturing efforts are greatly needed. There's no doubt about that. But also what has been shown conclusively over the past 50 years of studying coral diseases is that using only molecular or only histological methods are not capable of showing where in the coral host particular viruses, bacteria, and other microorganisms are present, and whether or not they're actually causing pathological changes in the tissues. So today we're emphasizing that combining disciplinary methods can address many of these data gaps. And kudos to those scientists who are already employing these methods in their work. But here we're calling for the increased practice of integrating histology and molecular methods in coral disease studies. So this is by no means all inclusive, but in the next few slides, I'll highlight some specific examples of combining molecular and histological techniques to study coral disease. And in fact, Esther and my lab collaborate to do this type of work. So this slide shows an example of immunohistochemistry. In this technique, a specific molecule of interest can be injected into a mammal whose adaptive immune system B cells will then secrete antibodies to the molecule, or antigens, that can be harvested. The location of that molecule in any tissue sample can then be found when the antibody is applied to and binds to it. A fluorescent or colored molecule then labels the antibody so that it can be seen using microscopy. So here in this light micrograph, you can see nuclei of cells dying by programmed cell death, or apoptosis. And they stain brown when the DNA is fragmented, resulting in damage to the nuclei and cell death. And these are preliminary results by graduate student Elizabeth McDonald, and they suggest that liquefactive necrosis lesions found in stony coral tissue loss disease are related to an increase in apoptosis. So in this next slide is an example of in situ hybridization, in which a single-stranded nucleotide sequence is prepared as a probe labeled with a colored or fluorescent molecule, and which binds to its complementary sequence present in the cells or tissues. So here we have the universal eubacterial probe, which binds to most bacterial phylotypes, is binding to what we suspect to be bacteria from previous Giemsa staining of a tissue section. And this is a project another student at George Mason, Samantha Cook, is working on. So correlative light electron microscopy is yet another method by which molecular and histological data can be integrated. Here is on the left is a transmission electron micrograph of deep sea mussel gill bacterial symbionts, which is overlaid with fish or fluorescent in situ hybridization micrograph, which is labeled with a methanotroph specific probe. So it's mapping the endosymbionts in relation to the host gill tissue. And on the right side is a parallel coral specific example. And in here, the top micrograph is a light micrograph showing granules in polyp calicoblasts of massive starlit coral Pseudastria siderea. And they stain pink with eosin or Giemsa stains, which indicate they contain proteins. And here they are considered to be the coral acid-rich proteins or carps that are attributed to forming aragonite crystals in corals. On inspection with fish, we were surprised to see that the bacterial probe also bound to these granules. And then when taking a closer look with transmission electron microscopy, it revealed that they in fact look similar in ultrastructure to primitive bacteria chlamydia. Finally, laser capture microdissection paired with microscopy can be used to remove cells from specific locations in tissue sections mounted on slides. The area to be isolated is selected, and then a laser is used to activate a film on a plastic cap to remove the selected cells. From there, the cells can be viewed to ensure capture and then processed for either nucleic acid or protein extraction. And this method can be used for taxonomic identification of suspected pathogens or symbionts, as well as gene expression and protein studies. And so as our laboratories endeavor to apply these methods to the study of coral disease, we often, of course, come away with many questions, prompting us to reach out to others for advice and collaboration. So with that, we'd like to close with this slide, reiterating that transdisciplinary collaboration is critical. And because of the complexity of diseases in corals, as well as other organisms, it's important to develop collaborations or mutualistic symbioses with other experts. Applying multiple techniques in field surveys and controlled laboratory experiments is essential to understanding the etiologies and pathogenesis of diseases, but no one person can undertake everything, and there's a niche for everyone in coral disease investigations. So that, we'd like to close, and thank you for your time.
Well, thank you both. Jen on the left here and Esther on the right. And it's time now, and await your questions later on. For the moment, it's time for Dr. Phil Dustan of the College of Charleston in South Carolina to put up his slideshow. While I tell you that Phil has conducted research in Florida, as you saw from his slides earlier, the Caribbean and the Indo-Pacific, most recently Indonesia, at scales that vary from the photobiology of zooxanthellae to the textural analysis of satellite remote images. And the title of his talk today is, Phil, where's your talk?
Well, I'm looking for it.
I'm looking at Jen.
I know. Hang on.
She's smiling.
You guys see it?
No, we see you now. We see you, but not your talk.
How about now?
Nope.
Nope.
Nope. We see you with a beautiful reef in the background, and it's obviously Indo-Pacific.
Hang on.
Did
Zoom
you
into the
lose it?
webinar. Tells me I should share.
There you go. The coral
Got it?
reef tourism pandemic. The floor is yours.
All right. Thank you so much. I'd like to really a big shout out here to Francisca and Chelsea and Judy for setting this up. I think this is a great idea. As the pandemic has come along, I've been involved in a couple of these, and they're getting easier to do, and they're actually getting more exciting. So welcome everybody. Big shout out to UWI there, home of Discovery Bay, and Cindy Hunter in Hawaii, and everybody else in between. What I want to talk about today is something I've been thinking about for a while, and you know, we live on an absolutely spectacular planet. As a matter of fact, it's the only planet we know of that harbors life. And life generates what we have here as a biosphere. We think in terms then of our wonderful blue marble. And when we sit back and we begin to look at it, we realize that life is not just universally distributed all over the planet. We find hotspots of biodiversity or more different kinds of species living in the tropical belt. And this has been attributed to the idea that the tropics are areas of high species diversity because they're biologically controlled. They tend to be older, they tend to be benign, they tend to be more predictable, and they tend to be an environment in which evolution can generate more different kinds of forms. And actually the interactions of those different kinds of forms is what enables those different kinds of forms to evolve. These are biologically controlled communities. And of those, reefs are the oldest, they're the most productive, and the most diverse of these ecosystems that we know of. And they are distributed in the tropics, as we can see on the top map here from Charlie Veron. We see the coral triangle in the very center, which is the heart of diversity. The second map, which is Reefs at Risk by Loretta Burke and her groups at WRI, we can begin to see the idea that some of these reefs are greater risk than others. And then the lower map, which was recently published by the Nature Conservancy in terms of the Atlas of Ocean Wealth, shows the dollar value, if you will, of coral reefs around the world. And right now it's somewhere around $36 billion a year. That's US dollars. And this, by the way, shows the distribution of humans on the planet. So humans have started expanding dramatically into this world. around 1750, around the time of the forming of this country here, where we are in America, you know, we had about 1 billion people on the planet. Now we're up to about 7. And the greatest rate of change has taken place from about 1950 to present. And the time period that I'm going to be talking about is between approximately 1970 and present day. So we've gone from about 4 billion people up to about 7 billion. And during that time, we have seen an increase in coral monitoring projects. And at the same time, we've seen a decrease in coral cover. And you wonder if they're correlated or not. And the reefs that we started monitoring were big, large structures that were emergent properties of all the organisms that lived on them. And Carey's Fort Reef, as Esther showed in the earlier photographs, now looks like this. It's basically a pile of rubble, and it's gone into what I would call sort of ecological ruin. In Bali Symphony Reef on the north side of Nanjengan Island, one of the bucket list dives in the world. In Bali, this reef was absolutely beautiful in 2014. And by 2017, it was nothing but a bunch of shards. In Dancing Lady Reef on the north coast of Jamaica, Discovery Bay, where modern coral reef biology got its beginning, or one of its starts with Tom Goro's lab, the Discovery Bay Marine Lab, had this wonderful reef that was really a magical place to go diving on. And I went back in 2013, and it looks like this. So you begin to look at all these reefs and their change, and it's like a war zone. And you really have to begin to ask yourself, is it time to imagine a world without coral reefs? And we know that the stressors are global and regional and local. They're nested, they're simultaneous, they're synergistic, they're catastrophic in their result. The largest global stress that we know of that we're just really beginning to see the rollout of is climate change from addition of carbon dioxide into the atmosphere. We've also added quite a bit of methyl chlorides and other molecules that change the composition of the atmosphere in many other ways, sort of promoting things like the ozone hole and acid rain and things like that. But anywhere you go on the planet, if you drop in the water and look at a reef, you can tell the symptoms of reef degradation. There are coral diseases, there are excess algal growth, there are sediments, crown of thorns in the Indo-Pacific, physical damages, and of course bleaching. And the paradox of all of this is the real interesting sort of enigma of reefs for years was, how do you get the highest expression of life in the sea, and it's thriving in waters that are devoid of nutrients and devoid of sediments, but they're really rich in energy. And life has figured out over the last 300, 400, 500 million years how to harvest that sunlight and trap and retain nutrients. And what we see now are reefs have very intense herbivory, they have very high filtration rates. We think of the reef as a wall of mouths, and these have evolved in a very predictable climate, the tropics. And we see symbiosis playing a very large role in this. You can think of a reef sort of as an economy. Industrial flow path analysis, industrial ecology looks at the imports into the reef and the exports coming out of it. We seem to, in industry, they think in terms of how these imports and exports influence the economy. In biology, we call this biogeochemistry. And this is why our planet is very different from all the other planets, because the living side of this two-box model keeps the Earth in a chemical disequilibrium and allows us to have things like oxygen in the atmosphere. At the same time, these ecosystems are processing energy according to the laws of thermodynamics. We see high-grade energy coming into the system. We see the metabolism that we would think of as community metabolism, production, consumption, decomposition, and then a lower grade of energy leaves the system. So energy flows through systems while the particulate parts, essentially the elements, can recycle. And the greatest example of this is in the symbiosis between corals and their zooxanthellae. So we have the animal that acts like a wall of mouths and traps nutrients and catching zooplankton. Its microbiome helps quite a bit in probably manufacturing all kinds of interesting vitamins and other molecules. And the algae sit there and chow down on photons. And in doing so, the two collaborate in polytrophy, and that results in enhanced growth of the colony, both in its tissue and its calcification. Another process that you'll see very common and sort of universal in ecology is that life will expand to fill the space into which it can. This is Vernadsky's theory of the biosphere from 1926. And so what we see is over this course of however many days, weeks, months, years, depending on the ecosystem, we go from early succession through to mid and late succession. And we see an enhancement in the amount of biomass present. At that same time, through this process, we see respiration steadily increasing and tracking biomass. We also see photosynthesis peaking in mid-succession and then winnowing down as this process, as this community fills up. So it's sort of like a large corporation. It becomes very productive when it's young, and then as it gets older and older and older, the amount of energy being used to run the system increases. And so the photosynthesis to respiration ratio of older systems is very small. As a matter of fact, Eugene Odom suggested that as a consequence of this, net community production or yield, what you can remove from a community, is large in young nature and small or next to zero in mature communities. And this is really critical when we come to reefs. So we can line all of these various communities up in a graph of community production versus respiration, and we can see that they all basically line up on this P to R ratio of 1. Actually, they're all slightly on the positive side of that, where the P to R ratio is slightly more than 1. And that's what they use to run themselves. There is no real energy left over in any mature system. They use all that energy to keep themselves operating. So let's sit back for a second and look at this. We have a system that has gone through its successional stage, and it's in relatively some sort of ecosystem stability or steady state. It's close to its carrying capacity. We see the inputs and the outputs are relatively constant. This is all superimposed on the energy of the system, and the system was in some sort of ecosystem homeostasis. In regulated communities, it's at this point that the biological forcing functions predominate. Competition, predation, herbivory, these density-dependent interactions are what drives and controls the community structure. And it gives rise to the idea of the Red Queen hypothesis, the biological arms race. And here's a perfect example of that. This is basically from Judy Lang and her legacy of watching corals overgrow one another. And here in the Dampier Straits of Raja Ampat, we can see all of this happening as different species of acropores are overgrowing each other. So think of a reef as an ecosystem, not a resource. The community is an emergent property of all the interactions of all the organisms there, and all those parts are needed for its sustainability. There is no extra stuff. There is no productivity that can be really harvested out of this system without influencing the system. Now, in the same time we've seen coral monitoring increase and explode throughout the world, we've also seen scuba diving explode throughout the world. And this is just the number of PADI certificates issued on an annual basis. And we're upwards of around 800,000 now. So from sort of a small group of people that were banded together in small groups like the British Sub-Aqua Club, we now have a raging business that is a multi-billion-dollar business going around the world. And you can think in terms of diving tourism supporting sustainable economies. And we talk a lot about that. And when you think about that, people are bundled into what I would call lodging units. If they go and they stay on land, they're in a homestay or a resort or villages or visiting a friend. And if they happen to be in a mobile floating lodging unit, they're on a yacht or a homestay liveaboard or something like that. So we have people living in places and moving around or being stationary. When these people come to these environments, they mostly live at a higher lifestyle, so they would like a Western infrastructure in this remote location. They've come to do some bucket list diving. They want to see the sharks or the mantas or the turtles or the big fish, but they'd like to have modern, safe diving. They'd like to have good guides. They want to be comfortable. They want to have wonderful meals, including high-protein seafoods. They want alcoholic drinks, air conditioning. They want the full Monty because they're on vacation. And this requires a significant supply chain infrastructure, tremendous amount of capital investment to make this work. And it generates a tremendous amount of human nutrient and operational waste. So the very adaptations that enable corals to thrive in these environments are going to make them vulnerable to the presence of humans. And the unintended consequence of tourism is to change the local ecology. We increase the runoff, we increase the amount of sewage, we increase the chemicals, we increase the physical damage, overfishing. We basically disrupt the local ecology. And you can break human impacts into three things. We can take, break, or pollute. It's very simple. We built this model a long time ago to try to understand some of this and to visualize it. And I'm not going to go into it right now because it's here. You can look at it later, but it basically tries to pick apart what people do. And you can roughly lump these into taking stuff, breaking stuff, or polluting stuff. So let's consider the hypothetical human impacts. And as G. Evelyn Hutchinson used to talk about, he'd said, environmental degradation is like being nibbled to death by ducks. So let's look at the impact of different kinds of ducks or people. A local might take a small amount, break some, pollute some, and have a total of, say, 60. Whereas a scientist might take a little more and break a little more and pollute a little more and have a sum total of 80. And underwater photographer, because they like to lie on the bottom and photograph stuff and they think they paid for this, they might break a little more. So anyway, we could theoretically at least put people into these categories. And so if we think about the growth rate of a population as being exponential and then density dependence added to that, we bring it down to logistic growth. And then we add another term. And that term is the sum of the impact of all these different kinds of tourists and people that are in that environment. And we could think of that as the net human impact sum. And that would be the sum of all the takes and breaks and pollutions from all the different human types and the sum of all of that. And if we took that and added it back into our logistic equation, it becomes a factor that essentially reduces the carrying capacity of the system. And so what it's going to do is it's going to decrease the carrying capacity as this system moves through time. Now, when we get into a situation where we have a relatively remote area that gets a huge influx in tourism, because now what we're going to do is instead of fish bombing and shark finning, we're going to have sustainable tourism. We see this explosive growth in visitors to the area, and it's also followed by an explosive growth of people to service that industry. And we now have, instead of 3 or 4 liveaboards, we have about 180, and instead of 5 or 6 homestays, we have 100. We see this exponential increase in tourism. And just to give you one example of that, if we think about increasing human nutrients, we think of a human as a day of activity, the human as a poop day. We can see that in 2000 in the Dampier Straits, we had about 9.7 million poop days, all the residents, all the visitors. Now, 18 years later, we're up to about 26 million poop days a year. It's a 374% increase in the amount of human waste going into that system. And so the reefs become nutrient sinks because they're starved for nutrients. They'll grab at anything. It's said that nitrogen is like crack cocaine for zooxanthellae. And the result is an explosion of degradation on the reef. For example, we see diseases downstream from these sources of pollution that 5, 10 years ago, nobody ever even saw or had heard of. So here we are, troubled and worried about the global changes that we see happening, mostly from global warming, but we also realize that we have to treat the resiliency of these systems, and we want to move towards having reefs that are locally more resilient, so hopefully they can withstand these global pressures that come along. So I would suggest to you that we step back for a second and think about this idea of sustainability. What we'd like to do is keep the reef in its steady state equilibrium, its so-called balance of nature. We need to do that by realizing that the reef first and foremost has to obey the laws of nature. We can integrate people into that nature being clever. We have to manage the people, not reefs. We can support communities and have sustainable tourism if we do it the right way. And in some respects, we have to think of each and every tourist as one of these little COVID creatures that's running around the planet and infecting people. And what we really want to do then is to manage the people as though they were a pandemic on the reef. So with that, I'd like to close and say I'm really appreciative that all you people are here. I think this is great. And thanks for listening, and I'll be happy to address any questions now or later. Thanks. Are we there?
Thanks, Phil. Judy will take us through the Q&A, and she will read the questions you all were posing in the Q&A, and then the plenary speakers will answer them for you.
Well, Francisca, all the questions in the Q&A are technical ones to you and I from people who are having trouble with their reception. So I'm going to ask, I'm going to go back to a question that came up for Esther, Dr. Peters, in the chat from somebody who perhaps wasn't logged on at the beginning and didn't know it was supposed to go in the Q&A. And that is, do you have any advice or information regarding coral histology procedures, additional to the wonderful book you already published, or if there are any workshops planned similar to the coral histopathology workshop held in 2005? So here's the next generation desiring to join you in the ever more exciting, expanding field of histopathology. Would you care to answer?
Yes, and I'm so glad to hear that people are interested and want to learn more. And we're trying to get more information out with some papers published, as well as offering other opportunities for training. Of course, in this time of the pandemic that we're all having to shift to online things, we have realized that a project that was started by Dr. Cheryl Woodley at NOAA a few years ago to develop a coral virtual microscopy system is going to be key to providing additional training. And for that, she collected a lot of slides. They have been digitized. They have been added to the archives at NOAA, and they have also now been put together by a graduate student of mine into a system that is using open source software, so that we hope that more people will be able to study them and learn more about how to interpret tissue sections. And that includes that we are going to provide additional scans of the tissues from the stony coral tissue loss disease samples that we hope to start working on shortly, and add those to the collection as well. Thus, we will have that part ready. And it has also been in the plans that I would provide course development working around this virtual microscopy system. I'm afraid I'm probably going to have to retire to do this, but maybe I will do it next summer. I don't know, but we will be trying to offer more training opportunities for people using this virtual microscopy system that the state of Florida is going to be funding the hosting of it, and we'll be sending out information on Coral List as those plans develop. Thank you.
So I do have another question here, which is that this is from somebody who has seen corals with stony coral tissue loss disease and remarks that they are very similar to corals with some very virulent types of white plague disease. And is there any histopathological evidence or molecular evidence as to whether or not they're the same or different? And of course, Jen can also contribute to that answer.
Well, we still don't know at the moment, but we have been working on this question. I received slides from one of my former graduate students who had taken tissue samples for study of white plague disease in the mid-2000s. And I'm going to be I've started looking at these slides to try to discern if there are any differences. And the slides were never looked at that time because he was busy working on the molecular microbiology. And so we then hope we have gotten him added into the microbiology work group that has been convened to study the bioinformatics coming out of the samples for molecular analysis. And hopefully we'll have a better answer about that by the end of the year. So yes, we still do not know at this time because no one else really studied the histology of the white plague. So we don't know, but we will hopefully find out.
All right, and here is another question for you. At the end of the talk, Jen mentioned 4 very high-tech sounding approaches to the study of coral histopathology. And it's realized that this is all work in progress and was slowed by the recent shutdown due to the COVID-19. All these graduate students, I guess, have not been in their labs for several months. But at the moment, which one do you think shows the greatest promise and why? And
well, of course,
I don't know who wants to answer that.
Esther?
Well, I would just say from my perspective, they all have great promise. And there are several of us who are working on stony coral tissue loss disease research who are using these combinations. And so I think they all show great promise. They, of course, are being used to study human diseases and veterinary diseases. And we have to move in this direction to study the coral diseases as well. Jen, do you have anything?
Sure. What came to mind is the recent pandemic, of course. It was the head of NIH that said they have, I think, 4 vaccines in development. And so you have, we're trying diversity approaches. And some of them are also dependent or integrated with one another. So we're hoping that they'll provide more insight into the disease and, again, in trying different methodologies, seeing which might yield the most useful information.
Thank you, Jen. That brought to mind the suggestion that was made earlier this year in the Florida Disease Advisory Committee meeting by someone that if the probiotic approach to discovering what can stop the stony coral tissue loss disease is successful, can you work backwards to identify the causative agent and that is responding to the antibiotic that the probiotic is producing? And that late in the disease, if I can make it double-barreled, are you even confident that that's what caused the coral to get sick in the first place?
Well, like any antibiotic, it depends on the specificity. Some are what's called broad spectrum, so they kill a number of different types of microorganisms, and some are more specific. So it would be dependent on that, but we're hoping with some of these more specific methods, we could, and as I mentioned, we have to move away from this idea that it's a single pathogen. It could be very likely that it is multiple types of microorganisms, or maybe some other external environmental factor that's causing programmed cell death in the corals. We don't know that yet. That's why we're trying to hit it from all these different angles. And there are many, many scientists using many different tools to look at this question. I'm trying to go back to the second part of your question. So you wanted to know if you'd be able to backtrack. So I think that would be hard to answer without more specific information, but certainly probiotics is yet another one of the tools in the toolbox that are being used in addition to other methodology to try to prevent the spread or colony mortality. So, and there's a lot of promise. They've seen a lot of success with it. So I think it's hopeful, but we still have not identified the pathogen. And if probiotics works, that's great. It's probably still important. We would like to know the cause of this disease, that we can take preventative measures in the future, or now even.
Well, thank you very much, Esther and Jennifer. Now I have a few questions for Phil, who seems to have disappeared from his background. Are you still there, Phil?
Oh yeah, I'm here.
Oh,
Just
you're
hanging
here.
out, just hiding.
Okay. I've had a question come in asking, how can you practically reduce the overfishing that you mentioned in that slide as being one of the factors adversely affecting coral reefs without unfairly penalizing the fishermen who are more easily displaced from their traditional occupations than are the more affluent capitalists and other financiers who are putting in the money for the resorts?
So in many places where I've worked in Indonesia, the fishermen become the people that drive tourists around in their boats. And so they have a role to play. And then the fishermen actually become very, very protective of the reef, because they realize that their income is tied to the reef. So what you want to do is you want to get the locals tied to that income. The other aspect is what we've seen is already the pressures that are coming on the reefs are sort of overbearing. So if you really want to have a reef, you have to back off from those fishing pressures. What they've done in Raja Ampat, which I think is really clever, at some resorts, they will buy fish from the fishermen, but only if they're pelagic fish. They won't buy reef fish. And there's a general moratorium on reef fish. Although if you go to the local market, you'll find reef fish. Reefs happen to be relatively productive, even though most of that really runs the system, but they can be drawn down quite a bit. The idea of marine protected areas and fishing places, you have some fishing spots, some spots that aren't fishing. I think the locals in many places are realizing that a fish in the ocean is worth more to show to people than it is to catch and eat. But then you have a shift in the diet of the people that live there. Nobody has a right to go out and kill all the fish. A lot of my friends go out and go fishing, and I say, hey, some of those are my fish. It's the commons, right? So leave some of them in the ocean. I'd like to leave my fish there instead of catching all of them. Although most fishermen will like to catch the last fish in the sea. So I think in some respects, what you have to do is limit the size of development. There has to be some control on this. If we just have raw capitalism, like what we're seeing in places like parts of Indonesia now, where there's this explosion of liveaboard vessels and explosion of homestays and resorts, sure, it's just going to drive the system into oblivion, and then the economy will disappear. So if we want to make a real, truly sustainable system, we have to go back to the laws of nature and obey the laws of nature. It's just like people now, this whole political thing about face masks. Oh, I don't want to wear a face mask. Oh, yeah, I want to wear a face mask. Oh, I don't want to wear a face mask. Oh, I do want to. No, I don't. It's a political thing. It's not. The virus doesn't care. The virus is just biology. Same thing with the reef. If we want to have reefs, we have to obey the biology, the principles of biology.
Well, thank you. That was a very complete answer. I have to admit that the places that I know of in the Caribbean where the fishermen are definitely helping with the sustainable tourism, or just the tourism, or even just the diving science industry. The amount of employment for them in those sectors doesn't equal to the number of fishermen who were previously fishing, and there's still a lot of excessive catching going on. So
Yeah, I would agree with you. In all probability, we have 7 billion people on the planet, which is probably about 4 times more than we need. And so we have overpopulation, and the overpopulation spills into the rape of ecosystems. I don't know a single ecosystem that doesn't really have some sort of pandemic disease problem. On land, we have the frogs, we have the bees, we have the birds, we have the sea otters. Everything is being diseased, everything is being overhunted, it's just being overrun by people. If we would like to somehow work on that, and maybe COVID is a time at which we can begin to get our priorities straight and begin to think about how can we lower the human population size over the next 3 or 4 generations. And the way to do that is to increase generation size and let it slowly work its way down. And there are lots of ways that we can do that. Or we can just sort of let it hang and slide and do what it's going to do, and it will crash eventually at some point. And that'll be the hard way down.
okay, Phil, we have time for one more good question, and I'm going to pull it from the chat from Tatiana Becker, who says, in Aruba, we have recently set up a study to monitor the effects of the presence of tourists on nearshore habitats, as we expect that tourist numbers will not immediately return back to normal after COVID-19. So there will be a gradual increase in disturbance with gradual increase in tourist numbers. And thus the disturbances on fish and sensitive species is expected to be lower. I am wondering if scientists and graduates on other Caribbean islands, and I wouldn't say why limit it to the Caribbean, would like to join us in our efforts to see whether similar patterns will be observed regarding fish and sensitive species like megaherbivores in nearshore habitats. And I know you can address that question.
Yeah, you know, we're actually doing this in Raja Ampat. We have the observations of the fish, but there's not enough gasoline. Gas is so expensive, it's hard to actually go out there. So we're looking at remote ways of doing that. But we're also sampling algae for stable isotope analysis. Because if the people disappear and the nitrogen signal disappears, we should see that in the integration of that signal into the reef organisms. So I would recommend that everybody go out. I know that in Hanama Bay, for example, Hawaii, they're doing some studies now. I would recommend that everybody that can get out and start to make observations on some aspect of the reef. And then when the tourists come back, try to really critically see what the tourists are doing. You know, try to individually trace tourists, see what they do, see how they behave, try to get an idea of what kind of carrying capacity can there be. In Indonesia, they realized there are too many people going to Komodo Island, so they increased the price of a ticket to $1,000. That way only the rich people can go. But maybe like up in the high Sierras where they have trails, there's a lottery. So the state of California will have a lottery that allows so many people to go to those areas. So maybe we can do something like that. We can begin to try to understand what this carrying capacity should be or could be, and gradually work our ways back to that.
Well, if we don't have any more questions, we are right on the nose at 4 o'clock. So I'm going to say thank you so very much to our 3 speakers today and to our sponsors. And that part of the screen has disappeared. So I will just have to read them to you. I can find them. We had funding for the remote meetings from the Company of Biologists. I knew it was a strange name, and I could see company down there on the top of that slide, but not the base, because they sponsored and made the Zoom meeting possible. And then TC there, Trade Wind Colors, sponsored the gifts for the plenary speakers and other trivia. So thank you to speakers and thank you to our sponsors. And I'm so glad you're still looking happy. And then the next slide will tell us what's happening now and tomorrow. So there's networking, which I think starts at 4:15. And there will be technical advances in reef studies talk session tomorrow at, and it's not coming up here, is that 1:45 to 2:45, Francisca? The Reef Bank Project and the integrative approach on the sustainable prospection of marine natural products, advanced technical diving for deep reef exploration, and customized medicine for corals. And then at 3 to 4, we will have, or maybe that's from 1 to 2:45.
Yeah,
and then
2:45 and then 3 to 4.
And then 3 to 4. That's not coming up here. Is it coming up on other people's screen? The time? Anyway, management and conservation plenary talks. Different topics, different speakers. Dr. David Gill will be evaluating the impacts of coral reef management. And Dr. Brigitte van Tussenbroek will talk about the impact of the sargassum brown tides on Mexican Caribbean coast. David's talk is more globally focused, and Brigitte is addressing a really serious environmental problem in the Caribbean at the moment. So thank you very much for attending, and stay tuned for the networking session if you've signed up for that.
Thanks for having us.
Yeah, thanks for having us. That was great.
You're welcome. All right, let's click end.
Was it just us or are people still there?